Welcome to Module 8, Basic Culture, Isolation, and Enumeration Concepts. In this session, we will examine the core methods used to separate microorganisms, estimate viable counts, and interpret culture plates with a quality-focused mindset. Culture work connects a physical sample to a reportable microbiological result. Every transfer, dilution, incubation condition, and counting decision can influence the reliability of the final conclusion. Isolation requires deliberate technique because a well-separated colony can become the starting point for identification, confirmation, or investigation. The goal is to obtain a pure culture that can be studied with confidence. By the end of this module, learners should understand the basic concepts of culture methods, isolation, enumeration, and plate interpretation. They should also be able to explain how technique, incubation, and record-keeping affect the credibility of microbiological results. The culture workflow begins with a representative sample and progresses through dilution, plating, incubation, counting, and interpretation. Strong microbiology practice means controlling the full workflow, not only observing colonies at the end. Different culture methods are selected according to the question being asked. Streak plates support isolation, spread and pour plates support viable counts, Broth supports growth observation, and direct inoculation can provide presence or growth evidence. Good culture results begin before the first transfer is made. Prepare and label materials, disinfect the work area, separate clean items from used materials, and confirm the planned dilution or plating sequence. The streak plate method progressively separates cells across the agar surface, so isolated colonies can develop. It is commonly used when a mixed sample must be purified before staining, identification, or further testing. The purpose of streaking is not simply to draw lines on agar. Each section should carry fewer cells than the previous section until sparse growth and isolated colonies appear. In the spread plate method, a measured volume of sample or dilution is placed on the agar surface and distributed evenly. This method is useful for viable enumeration when surface colony growth is required or preferred. Accurate spread plating depends on selecting a suitable dilution, pipetting the correct volume, and distributing the sample evenly. The plate should absorb the liquid before incubation, and all details must be recorded for traceability. The pour plate method mixes a measured sample or dilution with molten agar before the agar solidifies. Colonies can develop both within the agar and on its surface, which may affect their appearance and countability. During a pour plate procedure, microorganisms become distributed through the agar matrix. Gentle mixing, controlled agar temperature, and the avoidance of bubbles are important because they influence recovery and plate readability. Broth media support microbial growth in liquid form and can be used for enrichment, propagation, or presence/absence observation. Turbidity, sediment, surface films, and indicator changes may provide useful information, but broth growth alone does not identify the microorganism. Direct inoculation introduces sample material into a medium without a serial dilution or surface spreading step. It can be used for enrichment, growth demonstration, or presence/absence frameworks when the correct medium and controls are selected. Serial dilution reduces a high microbial concentration in controlled, stepwise stages. This makes it possible to obtain countable plates and then calculate the estimated concentration in the original sample. A dilution series must be logical, clearly labeled, and mixed consistently at every step. Use sterile tools as required. Mix each tube before the next transfer and document the dilution factor, plated volume, and plate identity. Dilution notation describes how much the original concentration has been reduced. For example, a dilution of 10 to the -4 represents a 10,000-fold reduction, while the calculation uses the reciprocal factor of 10,000. A colony-forming unit, or CFU, represents a viable cell or group of cells that can form a visible colony under the test conditions. One colony does not always originate from one individual cell because cells may exist as pairs, chains, clumps, spores, or aggregates. To calculate CFU per milliliter, multiply the colony count by the dilution factor and divide by the plated volume in milliliters. In this example, 86 colonies from a 10 to the -4 dilution plated at 0.1 milliliter give an estimated result of 8.6 times 10 to the 6 CFU per milliliter. For solid or semi-solid materials, the original sample mass and the initial homogenization step must be included in the calculation. A common mistake is to use only the plated dilution and forget the dilution created during sample preparation. A meaningful count requires a plate with sufficient colony separation and a colony number within the method-defined counting range. Plates that are crowded, overgrown, heavily spread, or difficult to interpret should not be used without following the applicable procedure. Not every plate is suitable for final numerical estimation. Plates that are too numerous to count, too low for strong precision, affected by spreading growth, or contaminated may require a different dilution, repeat testing, or a qualified report according to the method. Counting colonies is important, but their appearance also provides valuable interpretation information. Observe features such as size, shape, color, edge, elevation, surface texture, and opacity to identify unusual or mixed growth patterns. Multiple colony morphologies on one plate may indicate mixed sample flora, contamination, or incomplete isolation. Unusual patterns should be documented, compared with controls and duplicate plates, and investigated or repeated when interpretation is uncertain. Reliable microbiological results require traceable records, not just a number written beside a plate. At minimum, record the sample identity, preparation, medium, dilution, plated volume, incubation conditions, colony count, observations, and analyst details. Water microbiology requires careful sampling, controlled holding conditions, appropriate plating, and thoughtful trend review. A single result is useful, but recurring changes over time are often more informative for process and utility system control. Microbial counts from raw materials and in-process samples can support release decisions, risk assessment, and process control. The sample matrix, homogenization method, preservatives, and expected microbial load can all influence recovery and interpretation. Enumeration methods help assess the microbial quality of non-sterile pharmaceutical products and their starting materials. Results must be interpreted with product type, method suitability, neutralization needs, and the possibility of sample inhibition in mind. Food microbiology often uses homogenization, serial dilution, and plating to estimate microbial load or monitor hygiene. The sampling plan, product matrix, selected media, incubation conditions, and specification or trend limits all influence the final interpretation. Water-containing formulations, natural ingredients, and preservative systems can make cosmetic microbiology challenging. Effective sample preparation and neutralization may be necessary to recover microorganisms and interpret counts in relation to product risk. Many unreliable results arise from preventable technique or documentation errors. Incorrect dilutions, poor mixing, wrong plated volumes, improper incubation, damaged agar, and incomplete records can all compromise the final result. The most suitable method depends on the analytical question and the sample condition. Use a streak plate when isolation is needed, spread or pour plates for viable counts, broth for growth support, and direct inoculation or enrich